And with this endosymbiotic partnership in place, the descendants of the last eukaryotic common ancestor began to diversify, branching into the incredible spectrum of eukaryotic life we see today. To better study that diversity, scholars have been steadily refining what's commonly called the tree of life. Its branches represent all Earth's organisms and how they are scientifically categorized. These 7 supergroups branched away from LECA, forming into LECA's tree of life. Each supergroup contains distinct lineages, or clades, and within each clade, organisms are organized by the familiar taxonomic ranks: kingdom, phylum, class, order, family, genus, and species with subspecies occasionally recognized as well. As you can see, this clade, which derives from the Amorphi superphylum, is the Opistheconta branch. Once again, that's our branch. But it's still a bit premature for us to explore it, so we'll get into those details when they start to appear on the timeline. As for now, the timing is perfect for us to look at this major branch, Archaeplastida. This supergroup includes land plants, red and green algae, and a tiny, often overlooked lineage called the glaucophytes. Archaeplastida are defined by a unique innovation called chloroplasts. These are organelles that capture sunlight and convert it into chemical energy, which mitochondria then process into ATP, which is the universal energy currency of life. The origin of chloroplasts was a turning point in eukaryotic history. Roughly 2.1 billion years ago, a descendant of our last eukaryotic common ancestor engulfed a cyanobacterium. Gloeomargarita lithophora is that cyanobacterium's closest living relative. Therefore, Gloeomargarita is considered to be a living fossil. It was first described from the alkine lakes in Mexico back in 2007. It was then officially classified a decade later. Interestingly, unlike most bacteria, Gloeomargarita carries thylakoids. This, along with their other primitive traits, have become valuable clues when studying the origins of chloroplasts. Referring back to LECA's tree of life, the superphylum Picozoa appears to be the oldest living relative of the eukaryote that first engulfed the ancestor of Gloeomargarita. Through genetic and metabolic integration, this captured cyanobacterium gradually transformed into the first chloroplast. The process wasn't one-sided. Chloroplasts retain their own DNA and regulatory systems, sending retrograde signals to the nucleus to coordinate photosynthesis and stress responses. By about 1.9 billion years ago, this evolutionary merger had stabilized, giving rise to the Archaeplastida lineage, a branch whose descendants would eventually reshape the planet by oxygenating the atmosphere and building the green biosphere we know today. With our exploration of Archaeplastida origins behind us, we're now getting to the section of Earth's timeline when recently discovered archaeological evidence for eukaryotes begins to show up. And when we look for the first unambiguous fossils for eukaryotes, 2 dig sites rise into focus. One in northern China, the other in the Northern Territory of Australia. The Chinese fossils date to 1.63 billion years ago. The Australian fossils are slightly older, 1.642 billion years, just 8 million years earlier, a blink in geological time. Yet both discoveries push the fossil record of complex cells further back than we ever thought possible. The Chinese find, published in Science Advances in January 2024, was published by a team of Chinese microbiologists, joined by Harvard's Andrew H. Knoll, and released by the American Association for the Advancement of Science, the AAAS, founded in 1848, and publisher of the prestigious journal Science. It's another reminder of how the pursuit of knowledge often unites nations, no matter the politics of the day. But our focus now shifts to Australia, because this is where the oldest evidence lies, not only for early eukaryotes, but for the earliest known traces of purple sulfur bacteria and green sulfur bacteria. In the outcrops of the Northern Territory, rocks preserve an ancient world. Eukaryotic life thriving in tidal flats and lagoons, alongside microbial communities adapted to harsh sulfitic seas. The oldest of these layers is called the Blue Hole Formation. Fossils here preserve vesicles with delicate striations, fibrous or platy walls, and even woven textures. Some show opercula, which are tiny lids that opened and closed, like gates, to protect what's inside. Others display elongated neck-like extensions. These forms prove that by 1.65 billion years ago, eukaryotic cells had already achieved remarkable variety. It was once thought that the Tonian period marked what scientists called the Great Eukaryotic Expansion. But the evidence tells a different story. By the Tonian period, eukaryotes had already diversified into a wide array of life forms, shown not only by the fossils from Australia's Limbunya group, but also by the 1.63 billion-year-old multicellular eukaryotes of China's Chuanlingu Formation. The article's details do not end there. You see, in the very same Australian rocks, the team also found biomarkers, aka molecular fossils, of both purple and green sulfur bacteria. These microbes painted the seas of the mid-Proterozoic. Their pigments reveal an ancient ocean that was oxygen-poor, sulfitic, and permanently stratified. Eukaryotic algae struggled in these hostile waters, but purple sulfur bacteria and green sulfur bacteria thrived, forming layered microbial ecosystems across the globe. And so, the timeline speaks clearly. At 1.64 billion years ago, Earth hosted not just the oldest unambiguous eukaryotes, but also the earliest evidence of phototropic sulfur bacteria. Two milestones locked together in stone, reshaping our understanding of life's deep past. If you're interested in the nature of reality, then make sure you click the subscribe button so that you don't miss out on all the future uploads. And if you appreciated diving into the rock-solid evidence today, please also hit that like button.